Good afternoon. That's good afternoon in India, which I think it's still good morning there. But again, greetings from India. And thanks to the organizers for giving me this opportunity to present my little research work. So next slide, please. We know that we are all like whenever we talk about cancer, one of the major issues that come up is cancer relapse or recurrence. And that poses one of the major problems of cancer therapeutics, because so many times a patient has gone back home after treatment, after surgery, radiation, chemotherapy, no scars, nothing left, and the person apparently is fine. And then after 6 months to any time between 6 months and even 10 years, the person can come up with another tumor at the same place or at a distant site. And that tumor is then so aggressive that it's impossible for the person to be treated or cured and given another chance to increase their lifespan. So the question is, what causes this relapse? So when we were always thinking of therapeutics, we always wondered, like, what's happening? What causes this relapse? And we realized that there are certain cells which are sitting in that tumor, which are like, as you can see, that they are like dandelions in the backyard where you can cut the leaves off, but unless you kill the root, it keeps growing back. And it's like these persisting weeds which not only keep growing back, but they also kind of prevent the growth of other kinds of plants and in the garden. So while we were looking at what causes relapse, next slide, please, we realized that what was happening in the conventional chemotherapy is you have a tumor, which is, of course, a heterogeneous mass of cells. And when you're treating these cells, when you're adding any kind of drug, it's killing the bulk of the tumor cells. As you can see, the bulk tumor cells shown in blue here are the ones which are the dividing cancer cells. And the bulk tumor cells are obliterated, are removed by the chemotherapy and radiation and everything that's given. However, what remains is those little yellow cells which are there and which do not behave like the normal or the conventional bulk tumor cells. So these are a little different, and they are basically invincible because they are not killed by any kind of chemotherapy or radiation therapy. And they persist, and they're there. They're still sitting there when the patient apparently has been said that you are fine and you can go home and everything's going to be good. And then, that person comes back with a tumor later on because of these yellow cells which were sitting there. And these cells later have been very commonly termed as cancer stem cells or cancer initiating cells. And when it has been shown that when conditions become conducive again, this cancer stem cell, even a single cancer stem cell, can actually give back a tumor. So basically, this is what is leading to the recurrence or relapse of a tumor. So when we were thinking of designing therapeutics, we now changed our concentration on getting rid of these cells. Next slide, next please. So what we realize we need to do is to get rid of these yellow cells or the cancer stem cells. So once these cancer stem cells can be obliterated and we administer them along with conventional chemotherapy, finally the tumor would regress and we might achieve a more global cure for the patient, and the patient may not come up with a risk of recurrence in future. Next slide, please. So these cancer stem cells, this is what I'm going to talk about very specifically, are how we targeted these cancer stem cells using phosphodiesterase inhibitors. But just before coming to this, I'm just going to give you a little bit of background on how we arrived at this conclusion. Next. So these cancer stem cells basically are very resilient kind of cells, which have a very high self-renewal capacity. They are extremely chemoresistant. So the drugs which are given, even if first of all, they don't allow the drugs to enter. Even if they do, they are effluxed out because they have a very efficient protein pumps which efflux the drugs out. They are the metastasis rate is very high. The EMT, epithelial to mesenchymal transition genes, are very highly expressed in these cells. They also have a high rate of autophagy. So basically, they try to survive by using autophagy, and their apoptotic system is pretty low or suppressed, and most of the genes which support apoptosis are very minimally expressed. Next. So interestingly, what we found, this was a very interesting patient data that we found, where we use a particular assay called an aldiflor assay to assess these cancer stem cells, and this is represented as ALDH. So I'm not getting into the details of the technical part, but just a high ALDH means a lot of stem cells being there. So when we looked at a patient tumor, we found that there was almost 14.7%, an average of about ranging from 10 to 15% of cancer stem cells in patient tumors. If you look at a normal counterpart, you would probably find anywhere between 0.2 to about 1%. So that's a huge rise. But what was alarming was the panel on your right, which shows a CT tumor, which means tumor of a patient who has undergone chemotherapy. So once the patient has undergone chemotherapy, and the bulk tumor size has reduced, and the patient has, now they are just surgically removing the whatever little tumor mass is left, we find that the percentage of stem cells have increased radically. So which means that after the chemotherapy, when the bulk tumor cells have gone, these cancer stem cells are still persisting, and not only persisting, they're actually enriched in these patients. And by enriching these in the patient, it basically increases the risk of the patient in getting back the tumor, or facing a relapse after a few months or years. Next. So we wanted to understand these CSCs. We wanted to know, we then knew that we have to get rid of the CSCs. So all the conventional chemotherapy that has been given to cancer patients for so long is only targeted at the bulk tumor cells. But we realized that unless we get rid of the CSCs, it's actually never going to be a complete cure. So in order to get rid of them, we first needed to understand them. And next, please. In order to understand them, we carried out a microarray analysis to find out which are the genes which are actually making these cells so resilient and so resilient to chemotherapy, to radiation therapy, and how do they keep maintaining themselves and self-renewing and creating a reservoir of these cells in the body even after chemo and radiation. Next. So while looking at all the genes, we found that there is one gene which was extremely highly upregulated in the tumor samples, and more so in the chemo-treated tumor samples, and those were, that was SOX2. And SOX2 was the protagonist, which was actually helping the cancer stem cells survive and sustain themselves, even in spite of all the therapy that was administered. So next slide, please. So we've found out that if we can, we knew that SOX2 was helping these cells survive. So we performed silencing, a SOX2 silencing assay to see if anything improves. And yes, when we silence SOX2, interestingly, we found that after silencing SOX2, the other genes which were related to stemness, like OCT4 and NANOG, they were also reduced. ABCG2, which promotes chemoresistance in these cancer stem cells, was also reduced. And ALDH1A1, which is also a stemness gene, or which assesses stem cells, was also reduced. So we could reduce the stemness and chemoresistant genes. In addition to that, if we look at the bottom right-hand panel, you will see that this is a comparison between paclitaxel, which is indicated as PAX, and which is shown and which shows that paclitaxel actually does not reduce the levels of SOX2, OCT4, NANOG, etc., which is pretty high in the cancer stem cells. But if we silence SOX2 and add paclitaxel, which translates to a conventional chemotherapy along with a targeted chemotherapy for the CSCs, if we administer it together, definitely there is a reduction in the levels of SOX2, OCT4, NANOG, ABCG2, and ALDH. So definitely knocking down or silencing SOX2 was having very good effects, and it was helping us obliterate the CSC population. Next. I mean, it not only reduced chemoresistance, but it also, interestingly, when you silence SOX2, it is also increasing the sensitivity towards paclitaxel. So by silencing SOX2, we are also increasing the sensitivity towards a conventional chemotherapeutic drug. So in each and every respect, silencing SOX2 was promising. Next slide. This is another interesting result that we saw. If you just look at the control panel, this is a wound healing assay. And if you look at the control panel, this we can see that at 0 hours, we make a scratch in a plate of cells. And here we have used MDA MB231 cells, which are triple negative breast cancer cells. And when we plate these cells and we introduce a scratch, we want to actually look at how well the scratch is being, or the wound is being healed. And that will indicate how well the cells are migrating. So if you look at the control panel on the left-hand side, so at 0 hours, there's your scratch. And by 24 hours, most of the cells have migrated and they're trying to heal the wound. So the gap has reduced. However, if we remove the drug, there is an increased wound healing. So what does this indicate? This indicates that, if you look at this panel, you will see that basically the cells have the capacity to undergo metastasis because they are able to migrate and they're able to move in that plate of cells. Excuse me. However, if you look at the right-hand panel, which shows the cells which are being treated with paclitaxel, 0 hours, they all look the same. 24 hours, interestingly, there is no migration, which is very hopeful because we think that when you're adding a chemotherapeutic drug, the migratory capacity of these cells are reduced, hence they will not metastasize. But what was again alarming is when you remove the drug, the cells start moving again and they migrate again. So again, proving that once the patient has been treated and when the chemo and everything is over and the patient goes back home, the cells actually regain the capacity and they might lead to metastasis. And if we look at the genes which are related to EMT or, no, in the previous slide, please, the bottom panel. Can we go back to the previous slide, please? The previous one. Yes, no, the previous one. Sorry. Previous, the one before. Yes, we just stay here. The bottom panel, if you can just see, these are the MDA cells treated with paclitaxel. And while it's being treated with paclitaxel, we can see the EMT-related genes such as SLUG, snail, ZEB1, Vimentin. These are all downregulated. But once the drug is removed, which is on your right-hand side, we can see that the genes are again regaining their function. Therefore, the cells are migrating. But what happens if we silence SOX2? Next slide, please. So if we silence SOX2, interestingly, we can see that not only silencing SOX2 would prevent further migration. If you look at the absolute bottom panel of the cells where SOX2 is treated with paclitaxel and then the drug is removed, we can see that there is still no migration of cells, which means that If you just give paclitaxel, there is no guarantee after removal of the drug, the cells will start migrating again. However, if you silence it with SOX2 and then add paclitaxel, there will be a more permanent effect of non-migration, hence metastasis may be prevented. However, very interestingly, we found that even though all the other EMT genes were downregulated, there was one gene, which is TWIST1, which was constantly upregulated. So we wanted to find out what was happening. Next slide. So if you can just go next. There's a little bit of animation. Sorry about that. Yes. So in order to find out what's next slide. Why TWIST is upregulated? Because here we are seeing reduced migration, but we find TWIST1 still being upregulated. So we treated the spheres with specifically with SOX2. I mean, silence the spheres with SOX2. And next slide. Here we can see that when we silence SOX2 in the spheres very specifically, the TWIST1 is also being downregulated, right? So, which basically means that, so again, SOX2 needs to be silenced in the cancer stem cell population. I mean, if you silence it in the bulk tumor cell population, it will not help. But if you silence SOX2 in the cancer stem cell population, only then we can guarantee that all the genes which are related to EMT and metastasis are downregulated. Next slide. So definitely silencing of SOX2. Can you just go next 3 times? Yes, and yes. So if we silence SOX2, we can reduce stemness of these cancer stem cells, we can reduce chemoresistance, and we can even reduce metastasis. So somehow we have found out that, next slide, that if we can get rid of SOX2, if we can somehow silence SOX2 in the CSCs, we can obliterate the CSC. And once we can obliterate the CSC, we can provide a more global cure for the patients. Next slide. So how do we do this? How can we actually translate this and achieve silencing SOX2? I mean, we cannot do gene therapy every time in a patient. So how can we achieve this goal of silencing SOX2 in the stem cell of the patients? So when we did our microarray, as I told you initially, we found that there was a upregulation of SOX2 and ABCG2, and we have shown how modulating these two can reduce the stem cell population. However, when we next, if you can just stop if you can just we also found that there was a differential regulation of the CMP related genes and very interestingly, we found a very high differentially regulated PI3 kinase. And we know that the CMP-related genes and PI3 kinase are related to cell survival versus cell death. And we wanted to find out if this pathway was somehow being affected by, or is somehow affecting the stem cell population by modulating the SOX2 and ABCG2 levels. So next slide, please. As expected, we found that in the breast cancer stem cells, we find an increased phosphodiesterase expression, whereas an increased phosphodiesterase, as expected, would lead to reduced cyclic AMP levels. And this is the situation or the scenario which we found in patient tumors. So in patient tumors, we did see a high expression of PDE along with a reduced cyclic AMP expression. So we kind of drew a pathway which probably was helping these cells survive. And we derived at this pathway where a reduced cyclic AMP would reduce pKa, PI3 kinase, phospho-AKT, and phospho-mTOR. And when phospho-mTOR is reduced, this would lead to increased autophagy, and hence the CSC population would survive. And this is what we are observing in a tumor population. We are seeing survival of the CSCs. We find autophagy. So we derived at this pathway. However, when we carried out a Western blot analysis, yes, we found increased PDE. We found increased autophagy, which went from 9.6 to about 82%. But what we found different was pKa level, as we expected, was downregulated. mTOR was also downregulated. But somehow PI3 kinase and phospho-AKT levels were up. So this was a little surprising because we did not know why these were being upregulated. So next slide, please. Next. So next, here we now added rolipram to see what was happening. And once we added rolipram, we found that the PDE, the PDE level was now reduced. The CMP levels were now upregulated, as a result of which PK, PI3 kinase, for phospho-AKT, which were all supposed to be downregulated, were now upregulated. So when phospho-mTOR is upregulated, autophagy was downregulated. So we see now autophagy coming down from 89 to 32%. And this is actually leading to a non-survival strategy of the CSCs. And we presume here that pKa here is playing, next please, pKa here is playing a direct role, and it is bypassing the PI3 phospho-AKT and directly affecting phospho-mTOR, downregulating, upregulating phospho-mTOR so that the rate of autophagy reduces. So when rolipram is added, we basically can see that the autophagy levels come down. The cells are now in a non-survival strategy. So finally, are the cells dying? Next slide, please. Yes, we notice, next please. We finally see that yes, downregulation of autophagy is also correlated with an upregulation of apoptosis, as seen by all the apoptosis markers. Next slide, next please. You can definitely say that when rolipram is being added, there is some kind of a deregulation. We are inhibiting the phosphodiesterases, as a result of which phospho-mTOR eventually gets upregulated, leading to downregulation of autophagy and upregulating apoptosis in these cells. And upregulation of apoptosis mainly occurs by an stabilization of the Becklin BCL complex, which proves that it is probably an autophagy-induced apoptosis which is taking place. So finally, we are able to carry out apoptosis and bring about apoptosis in these cells, and we are able to obliterate them. Next, please. So finally, we can see that SOX2, next, if you can just, yes. So finally, what happens if we add rolipram, we have shown that it is not only leading to downregulation of phosphodiesterases and modulating the CMP pKa pathway, leading to apoptosis. And finally, this also downregulates SOX2, as we can see that it can also downregulate SOX2, finally leading to a complete obliteration of the CSC population. Next. Yes, so finally, together we can obliterate the CSC population, which we are extremely keen on achieving. Next. So with that, I would just like to thank all my research scholars, the patients who have donated their samples for which I could carry out the experiments, my collaborators, funding agencies. And finally, next slide. Yeah, thank you everyone for giving me this opportunity, and thank you for bearing with me. Thank you.